In this video, we will discuss about the immune cells and the tumor microenvironment from the pancreatic ductal adenocarcinoma point of view. So far, we know that the stroma or the tumor microenvironment that is formed across the pancreatic cancers has immunosuppressive properties. And from the immune cells point of view, they are mainly composed of the macrophages, and that too, this M2 type macrophages, which has a tumor proliferation properties. And the other type of macrophages, that is M1 type macrophages, which has a pro-inflammatory properties, are low in this tumor microenvironment of the cancer cells or pancreatic cancers, and it will also affect the innate immune response. And we'll start with this NK cells, and NK cells can be correlated with the cytokines that are found in tumor microenvironment, and cytokines such as interferon gamma, IL-2 are found to be low, and whereas the cytokines such as interleukin 10 are found to be high. And this indicates that this NK cells has a regulatory role, that is, they are present in the tumor microenvironment, but they don't have a cytotoxicity effect, and they plays a role towards tumor proliferation. And at the same time, for the CD8 plus T cells, for this T cells, what it will does is in the tumor microenvironment, the regulatory T cells are found to be high, and these regulatory T cells will prevent the activity of the CD8 plus T cells or CD4 plus T cells. That is, it will suppress the activities of the cytotoxic T cells and the helper T cells. And from this is CD8 plus T cells is from the point of view of adaptive immune response. And apart from that, the cytokines that are released, such as this TGF beta or CXCL13. So the CXCL13 cytokines will attract the B cells, and more importantly, there's PDL1 plus B cells, and the PDL1 plus B cells then further helps in the suppression of this immune response, such as by interacting with the CD8 plus T cells to suppress the cytotoxic effect of the T cells, and thereby helps in the tumor suppression and proliferations. And apart from that, the other things that we have already covered in the previous video is about this collagen, HAMMP and TIMPs, and this cancer-associated fibroblast that release interleukin 2 and interleukin 6 that will inhibit this, also helps in this NK cells inhibitions. And these regulatory T cells usually inhibit these cytotoxic T cells. So this is in general how this tumor microenvironment of the pancreatic ductal adenocarcinoma interacts with these immune cells. And there are several strategies that has been used based on these informations, such as using anti-PD1 or immune checkpoint blockade therapies, along with those chemotherapies or along with the other targeted therapies to improve the efficacies, as anti-checkpoint inhibitor therapies will neutralize this B cells that are attracted towards this cancer cells by this CXCL13 cytokines. Or one can also target the CXCL13 cytokines that will prevent PDL1 plus B cells to be attracted towards this tumor cells. The other strategies that people have seen is that if these CD8 plus T cells are present within the infiltrate within the tumor microenvironment, then it can have the cytotoxic effect against the tumors, and at the same time it will activate the other immune cells such as NK cells by providing the antigen informations to the dendritic cells. So this is in general how the cancer cells and immune cells interaction plays a role. So if you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. about this immune cells, tumor microenvironment, and the cancer associated fibroblast interactions. That's all for this video. Thank you for your time.